neuropathological features of COVID-19, our work was really in a wonderful collaboration with Isaac Solomon at the Brigham. We were able to do autopsy studies both for full body as well as brain. And in brain studies, we looked at individuals that had consecutively passed away from COVID-19. So these are individuals that did not necessarily have neurological manifestations, but had died from largely the respiratory complications. These individuals all had 100% had hypoxic ischemic injury that you could identify in the brain. What was interesting and unique a little bit about COVID-19, and it's different than what we see many times in other neuroinvasive viruses, that patients with COVID-19 had very little to minimal neuroinflammation, so inflammatory cells that were inside the brain space. We were able to identify with our collaboration with Pardes Speti and the Broad very low levels of viral RNA for SARS. This was done by quantitative PCR, and we looked at multiple different sections of the brain. This study was the one that was published in the New England Journal. Most studies are not able to identify protein or an infected cell, with the exception of one study which did identify some of the cranial nerves are infected with SARS, and there's some scattering of cells in the brainstem that do have SARS immunohistochemical positivity. What was interesting about this study that correlates with the study that we published in the New England Journal is essentially there's really minimal pathology that seems to be associated at least with this viral presence. Other work that we'll be continuing on is to also look in with collaboration with Bruce Fischel at the Martino Center, as well as Brian Edlow here at Mass General, is to how do we use imaging, so MR imaging at an ultra high resolution to target our histopathological diagnostics.